Welcome back to a new video session from Biology Boost-Up, and in this video we going to learn about what is inhibition and the role of inhibitions on enzyme activities. So now let's start. Inhibition of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemicals shut off enzyme activity, the process is called inhibition and the chemical is called inhibitor. The inhibition are two types. First, the reversible inhibition and second is irreversible inhibition. If I talking about the reversible inhibition, it is the inhibitions occurs when an inhibitor form weak chemical bond with the enzyme, and the reversible inhibition again subdivided into four categories. First, the competitive inhibition, second is uncompetitive inhibition, third is feedback inhibitions, and fourth the non-competitive inhibition. In case of irreversible inhibition, the inhibitor form a strong chemical bond with the enzyme. As a result, the inhibitor permanently inactive the enzyme. Now let's talk about the competitive inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzymes is known as competitive inhibition. In this type of inhibition, due to its closely structural similarities with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzymes. As the substrate cannot bind, as a result, enzyme action declines. In these conditions, the Vmax stay constant. The Km increases, but the inhibitor does not affect the turnover number of enzyme. As you can see here, the competitive inhibitor is often a product of the enzyme catalyzed reaction. When a lot of product is made, the product would compete with substrate for the enzyme's active site. The competitive inhibitor is able to fit at the active site. When it is there, the substrate is unable to bind to the enzyme and the desired reaction does not occur. Here, the inhibitor as well as the substrate both compete for the active site. In case of uncompetitive inhibitor, an inhibitor also bind at a site distinct from the substrate. However, an uncompetitive inhibitor will bind only to the enzyme substrate complex. As a result, both the Vmax and Km decreases. Now let's talk about the non-competitive inhibition. The inhibitor binds to the enzyme at a site other than the active site. Inhibitor bind alter the enzyme's three-dimensional configurations and block the reaction. Inhibitors bind to the either free enzyme or enzyme substrate complex. Here the Vmax decreases and Kmax stay constant. This diagram saw an inactive form of an allosteric enzyme. A non-competitive inhibitor is bound to the regulatory site The active site is thus changed and the substrate cannot bind to the enzymes. Now this is the most important topic in this video session, the allosteric modulations or feedback inhibition. It is a type of reversible inhibition found in allosteric enzymes. The inhibitor is non-competitive and is usually a low molecular intermediate or products of a metabolic pathway having a chain of reaction involving a number of enzymes. It is therefore also called end product or feedback inhibition. An example of feedback mechanism of inhibition is stoppage of activity of enzyme hexokinase by glucose 6-phosphate, the product of reaction catalyzed by it. As you can see in this diagram that there is a substrate which is produce product A by the help of enzyme 1. Then this product A produced product B by the help of enzyme 2, and this product B then again produce product C by the help of enzyme 3. Now this product C inhibits the activity of enzyme 1. As a result, the product A will not further again produced by the help of this enzyme. This is a chain reaction. Now look, what is the turnover number? The turnover number of an enzyme is number of substrate molecule converted into products per unit time when the enzyme is fully saturated with the substrate. This turnover number of an enzyme dependent upon active site. Carbonic anhydrase, which is present inside the RBC, is one of the fastest enzyme known with turnover number 6 lakhs per second. Thanks for watching my video. Thank you, friends.